right.
You are ambiguously Middle Eastern.
So
This area.
we are going to start today with the last section of our. So we're going to finish with our last section of sort of genetics here. So what we have left, we've already talked about Mendelian genetics and Punnett squares, inheritance, pedigrees. So what we're going to talk about from here now are just a couple other topics related to genetics, mostly focusing on genetic technology. But before we get to that, we have to talk a little bit about population genetics. So one thing that ecologists and geneticists study is not just the genetics of individuals, but also the genetics of an entire population, an entire group of individuals. Okay? The study of gene frequencies and the study of the inheritance of those alleles through a population is called population genetics. And so we need to understand some definitions to understand population genetics. When we talk about a population, we're talking about a group of individuals of a single species living in a specific location. Now, if we're studying sort of the the population genetics in squirrels in the northeast of New York State, we would only include the squirrels within that region as we study their genetics, and not the same species of squirrels living in California, because obviously they're not going to be reproducing together. And so we're only looking at a specific population that might be sharing genes. When we talk about the gene pool, we're looking at all of the combination, all the various alleles within that population. And then when we talk about gene frequency, we're saying, okay, how common is this certain allele within the gene pool of this population? And so one scientific principle that's commonly used in population genetics is this concept of Hardy-Weinberg equilibrium, or the Hardy-Weinberg principle. And the Hardy-Weinberg principle says that the gene frequencies in a population will remain stable under certain conditions. That the frequency of certain alleles is not going to change very much through a population. But for a population to reach this Hardy-Weinberg equilibrium, it has several stipulations, several conditions that must be met for the population to be at equilibrium. So one is that it has to be a large population. With small populations, there can be large changes in gene frequency because there's a very small gene pool. There has to be just random mating, no sexual selection. There has to be no migration of individuals coming into the population or leaving, because obviously that would change gene frequencies in that population. There has to be no mutation, no natural selection. So there's a whole bunch of conditions that must be met in order for the population to exist at this. So, and often that's not the case. And so Hardy-Weinberg equilibrium is sort of an idealized model. Okay? And we can use this concept to allow us to sort of predict what the probability of individuals in a population of having certain genotypes. So it comes with an equation that you will have to be able to use. Okay? This is sort of definitely an AP Bio sort of topic and not a Regents topic. You'll never have to know anything about Hardy-Weinberg equilibrium on the Regents exam, but you will on your test. So the equation for Hardy-Weinberg equilibrium is p squared plus 2pq plus q squared equals 1. So let's look at what these letters actually mean. So p and q are two important things that you'll have to know. So this would be something you would be given. So the p represents the frequency of the dominant allele for a certain trait. Q represents the frequency of a recessive allele. So these are given in decimals, okay? 0.70, 0.25, or whatever. Okay? So then if we have the frequency of the dominant and recessive allele, if we have P and Q, we can then use them in this formula to calculate the frequency of homozygous dominant genotype, heterozygous genotype, or homozygous recessive genotype. P squared is the frequency of having the homozygous dominant genotype. 2pq is the frequency of having the heterozygous genotype, and q squared is the homozygous recessive genotype. So we'll look at an example here in a minute. All right, so let's look in this problem. You don't have this page, but you just look at it. We have these insects. We're looking at a gene for the body color of these insects. There's two alleles, dark and light. Okay, the dark colored allele is dominant, represented here by capital A, to the light colored allele, which is lowercase a. So here I'm giving you the frequency. So in this population, which is at Hardy-Weinberg equilibrium, the frequency of the big A allele is 0.3. 30% of the alleles are big A, and 70% of the alleles are little a, the recessive allele. So, given that information, we can then figure out the frequency of having a homozygous dominant genotype. So, what is the frequency of having big A, big A? Anyone figure it out? Dean?
0.9.
No? But you're close. Correct. So, P squared, which is 0.3. 0.3 times 0.3 is 0.09. So what that means is 9% of this population will have the homozygous dominant genotype. What percent will have a heterozygous genotype? Carter? Okay. Or 42%, yep. So 2pq, 2 times 0.3 times 0.7 equals 0.42, which is 42%, big A, little a. And what percent would be homozygous recessive?
49.
Yes, 49%. So again, in this population, there is a large percentage, almost half, get the homozygous recessive genotype. And so the dominant genotype is not always the most common, right? In this case, you would expect there to be relatively few recessive, or often people think that, but actually because the frequency of the recessive allele is so high, you actually have a large number that are homozygous recessive. So this is how we use this formula. As you get more advanced, like in your AP biology, you may be given some of this information and have to then derive what P and Q are, but it's the same, just using that same information. This is all you'll have to do for our purposes. All right, so let's talk a little bit about modern genetic technology. So there have been huge advancements in the last 20, 30 years in terms of how we understand DNA and genes and how we apply modern technology to our genes. So genetic engineering, biotechnology, these are terms that sort of we use to talk about this, this field of science. Often when we talk about genetic engineering, we're referring to altering the genetic code of an organism. And so here's one type of genetic engineering. This is called gene splicing, where it says these simplified steps. So we know that our genes, our DNA, is a series of A's, T's, G's, and C's. And a gene is basically has a code that tells our cells how and when to produce certain proteins. Our genes are codes for how to build proteins. And in this example, let's talk about insulin. So 40 years ago, a person was a diabetic that needed insulin injections. That insulin came from pigs, came from slaughtered pigs. They would extract insulin from the pancreas of those pigs. They package it, and that's what people would inject, pig insulin. It worked okay. It wasn't as effective as human insulin. There were a lot of problems with rejection, with allergies to it. So it worked, but not ideal. Since then, scientists have used genetic engineering to produce insulin in a different way. And they've done it using these gene splicing steps. So to do this starts with extracting the DNA that you are interested in. So basically you can use any tissue sample to extract DNA. Even a small sample of tissue can extract the DNA from the nucleus of those cells. There are ways of amplifying the DNA to produce more of it. And then scientists have figured out how to basically cut out one specific gene from that DNA. So they know where the human insulin gene is, the gene that tells our body how to produce insulin. They've identified it. They can go into the human genome, we call it, and cut out the insulin gene. To cut out genes, they don't use mechanical tools, they use chemical tools. The tool that they use to cut DNA is called the restriction enzyme. It's an important word. It's commonly asked about on the Regents exam. Restriction enzymes are enzymes that scientists have identified from different organisms that cut DNA at a very specific place. Scientists have restriction enzymes that always will cut at a certain sequence of DNA. That's what's shown in this lower left-hand corner. So we have a double-stranded DNA, and a restriction enzyme, when added to it, will cut it at a specific place. We'll always cut it at this specific sequence. So scientists have found the human insulin gene. They can cut it out of the human genome, and they take that gene, they can then actually insert it into another species. What they've done is they've taken bacteria DNA. They've cut open the bacteria DNA and basically pasted in the human insulin gene. So now we have a piece of bacteria DNA, but spliced within it is the gene to produce human insulin. We put that gene back into a living bacteria, and now that bacteria can live, but it can also do what? Produce insulin. And we know that bacteria can reproduce asexually, copying themselves many times. So scientists can then grow a huge culture of these genetically engineered bacteria, but they're all now able to produce insulin. And they can extract that insulin from those bacteria, and that's where we get insulin today. That insulin is it's human insulin. It's being produced by a bacteria, but it's identical to the insulin we all produce in our pancreas. So we've taken that gene out of humans, put it in bacteria, and now these bacteria can produce insulin. They've done the same thing with human growth hormone and with other hormones. Cut it out of a human, put it into a bacteria, grow the bacteria, extract the product. Here's a diagram showing you. Okay, you see the two cells, the human cell, the bacterial cell, find the gene, cut it out using a restriction enzyme, get the bacterial DNA, cut that open, paste the human gene into it, and now we have what's called recombinant DNA. DNA that has genes from two different species, bacteria and humans. Put it back into the bacteria and let it do its thing. So that's one form of genetic engineering, that's gene splicing. We can now sequence DNA very quickly. It used to be a very painstaking process. There was a race to catalog an entire human genome. It's called the Human Genome Project. There are competing groups, competing labs, trying to identify all of the bases in a complete human genetic material. And these labs had different ways of doing it, and they were sort of racing each other to do it. And eventually, they did it. Today, we have computers and we have technology that can do it very, very quickly in an automated way. And so we have DNA sequencing equipment. We can even actually make our own DNA sequences. That's called synthetic DNA, where scientists can sort of put together A's, T's, G's, and C's in a specific way to produce a gene that they want to produce. They do amazing things with DNA today. So by sort of this process of cutting and pasting genes from one organism to another have allowed us to start creating transgenic organisms, organisms that have DNA from another organism put into them. Those bacteria we were just talking about, they are transgenic. They have genes from two different species. So that was sort of the first avenue in which scientists started using genetic engineering for sort of applications that were helpful to humans. They've now taken this further. We can create and have created transgenic animals as well. We can take, for example, the gene to produce human immune cells, put that into mice, and then the mice can produce those cells. We could take genes for human diseases, like heart defects, put them into mice. The mice develop those heart defects, and then the mice are used for research purposes, to try to come up with treatments and cures for certain genetic ailments. We can take genes that produce certain medicines and put them into cows, and the cows will then produce these products, these medical products in their milk, and the products can then be extracted, basically using living organisms as factories to produce certain compounds. This is done quite frequently in agriculture with crops. For example, some plants naturally produce insecticides, chemicals that kill insects. It's a protective strategy to prevent insects from eating them. Well, if we take that gene out of one plant and put it into a crop like soybeans or corn, we can reduce the damage that insects would do to those crops. And so scientists have done that. They've taken these genes to produce insecticides, put them into our crop plants so that insects are less of a pest to them. These are soybeans. So Roundup Ready soybeans. You know what Roundup is?
Weed killer.
It's a weed killer. It's a really strong weed killer that basically kills almost all plants. You might have it at your house and a body might spray it in the cracks of your sidewalk or around your driveway to kill any plants or weeds. So Roundup is a strong insecticide. Now, you can't just spray Roundup in your field of corn because it'll kill your corn and the weeds. However, scientists have found a gene that makes plants resistant to Roundup. They've taken that gene, inserted it into crops like soybeans. So these are Roundup Ready soybeans. So you have a field of soybeans growing. Farmer can then spray Roundup throughout the whole field. The soybean plants won't be affected because they have this gene in them, but all the other weeds will be affected. Okay, and so these are transgenic plants. This is golden rice. It was engineered rice that produces more vitamin A than normal rice. Vitamin A deficiency leads to blindness in children that are malnourished and not getting enough vitamin A. And so by creating this strain of rice that has increased vitamin A, it's been able to drastically reduce the incidence of some of these diseases in children in developing countries. So there's lots of ways scientists have used these transgenic organisms. Now there are controversies. Some people feel that humans should not be mixing genes from two different species. Some people feel that there are dangers involved in allergies or in these genes spreading or in unintended consequences. So, you know, if you ever read about GMO foods, you know what GMO stands for? Genetically modified organisms. That's what we're talking about here. These are all genetically modified organisms. And so some people feel we should label all foods that come from animals or plants that have been genetically modified. Lots of food groups, producers would say we shouldn't, but you know, there are definitely things to think about in this. Our technology has been increasing drastically. You may have heard in the news of CRISPR-Cas9. CRISPR is a genetic tool that allows for the more effective and easier editing of genes within an organism. Using this technology, scientists are making progress in fixing genetic defects in actual tissues of cells in living organisms. The idea is that someday using this technology, we could take corrected genes, so cystic fibrosis, for example, caused by a single mutation, one incorrect protein produced in the lungs, leading to a often fatal disease. Scientists think that there is a possibility we could deliver corrected genes into the lung cells of people with cystic fibrosis using this technology to basically fix those cells inside of a person so they no longer have this genetic defect. So we talked in the past about sort of the ethics of whether or not we should modify genes and where do you draw the line at correcting a disease versus enhancing an individual, and there's a lot of things to think about there. But the idea is that we can edit those genes inside of a person or an animal. Cloning. So we can clone organisms. This happens very commonly. A clone is basically a genetic copy of an organism. So oftentimes today we clone tissues. We can clone heart tissue from a certain person, but we can clone entire organisms. The first and one of the most famous cloned animals was a sheep. Her name was Dolly. In 1997, she was produced. Many mammals have been cloned since. Some people have claimed to have cloned humans, although they have not provided proof that they have done that. You could send a sample of your dog's DNA to South Korea, where they will, for over $100,000, clone your pet and send you back a puppy that is a clone of your dog, an exact genetic copy of your dog. The technology is not that complex. This is the way Dolly was created, and this is the way animals are still cloned. So you have the animal you want to clone. Let's imagine it was a puppy. This would be like your puppy you want to clone, or your dog. No, your old dog that you want to clone. So you take a sample of cells from your dog's cheek. You get a cell. You take the nucleus out of it. Get rid of the rest of the cell. So you have a nucleus from your dog. It has your entire dog's genome in it. All the genes that made your dog your dog. You then take an egg cell from a female dog. The egg cell only has half of the DNA in it. It's a gamete. But we get rid of the nucleus. So we have an empty egg cell. We put your dog's nucleus into this empty egg cell. And then we shock it with a little bit of electricity in a petri dish. That will stimulate it to start to grow from a single egg cell with your dog's nucleus in it into 2 cells and 4 cells, and it forms an embryo. But that embryo is a genetic clone of your dog. They take that embryo, implant it into some surrogate dog's uterus, and that dog's pregnant, but the puppy inside is not her genetic offspring, it's your dog's clone. She gives birth to that after a little while, and that puppy that's born is a genetic clone of your dog.
Wasn't there like something where they had like shorter life expectancies?
Yeah, to produce these clones, oftentimes they have to produce like a bunch of embryos, and many of them don't actually implant and develop normally. So there is some trial and error in this process. It's one of the reasons people have not ethically thought that it should be done with humans. But the steps are the same. You could clone a person using those same steps. Take a nucleus from Meg, put it into an empty egg cell from some other woman, implant that, shock electricity, implant it into some other surrogate mother. She would give birth 40 weeks later, but that newborn baby is Meg's clone. It'd be like, now, would it have Meg's brain and her thoughts and her feelings? No, it's another individual. So sometimes you'll think like it's a clone, and but it's just like an identical twin that happens to be separated by 15 years. Yeah, it's like staring at the All right. So our last topic to talk about is a process. I wanted to actually do the whole lab on this. I don't think we're gonna. I think we'll do this as a demonstration next week. Electrophoresis. We have the equipment to do it here. But electrophoresis is a tool that's used oftentimes for several different reasons. The basics are that electrophoresis separates a mixture of molecules by size by using electricity to draw these molecules through a gel. And so it's done using DNA very frequently, where they take a sample of DNA, they run it through a gel, and because the DNA is negatively charged, it's attracted to the positive electrode, and so the DNA starts to move. So the way you do this is a little gel tray. You pour a liquid gel into this tray to give it its shape. The gel solidifies. It's like Jell-O, but it's clear. And then you take that gel that has these indentations. They're called wells, and you put a sample of DNA in the wells. You plug in these electrodes, and it runs electricity through a solution in here, and the DNA starts to migrate from negative to positive. Now, small pieces of DNA can work their way through the gel very quickly, and they go far. Large chunks of DNA can't go through as quickly, so they don't go as far. So it produces a pattern of bands in the gel. You stain it, and you could see the bands. And then you could compare the bands to see how these DNA samples vary. This is used to create DNA profiles for paternity testing or for forensic research to see, if you have a sample of DNA from a crime scene and then you have several suspects, if you make electrophoresis, a gel electrophoresis of their DNA samples, you could compare them and see which person matches the DNA that was found at the scene of the crime. So again, you see the DNA is cut up using a restriction enzyme. The samples are put into the wells, electricity is run through them, the bands are stained, and then you could compare those stains. So here's an example. Here we have a mother and a child, but we don't know who the father is. So we can create a DNA profile of these individuals. There's 3 possibilities for who could be the father of this child. If only they had this in Westeros, it'd be perfect. So we know if this is the child's DNA profile, that every band in the child must have come from either the mother or the father. So we have the known mother, we have the child, and then we look at, okay, well, where did these bands come from? If we look at in the child this first band, well, the mother doesn't have one here. So this must have come from the father. So this father, he doesn't have a band here. So he couldn't have been the father. This father does. This possible father does. So either of them could be the father. Continue down. Well, this one came from the mother. From the mother. Here, these next two came from the father because they're not present in the mother. Okay? Well, this one could have come from this possible father, or this one is the one that does it. So this is the only father that could have provided all three of these bands to this child. So this little guy, he's the father. So it can be used not only for paternity testing, but for lots of other purposes as well. All right, any questions about electrophoresis or any of our other things we've been talking about?
Wait, don't get rid of it yet.